a little bit confusing. Um, but you have HLA and then you have the locus or the gene. So if you're looking at like DR beta or A, B, C, then you'll have the allele group, which will be the first two spaces, or sometimes it's written just with a 1 if there's a 0 in front. So you might have an A2 or a B3. And then you get into the different proteins. So it will be a similar type, but the protein will be slightly different if it when you're looking at the third and fourth field. And you can determine this by molecular typing. In previous days, they used to do all HLA typing by serology, and you would only have the first two fields. But now when you're doing molecular typing, you will have at least 4 fields. So, and this is very basic. The gene is found on chromosome 6, and you have 3 classes of HLA genes. So you have class 1, which is ABC, class 2, DR, DQ, and DP, and then class 3. You don't really hear too much about class 3, and most things will only focus on class 1 and 2. Especially in like transplant, we only really care about 1 and 2. And then if you're talking about transfusion, we only care about class 1. And the reason we talk about HLA in transfusion is for platelets. Platelets do have class 1 antigens on them. So if a patient has antibodies, they may be platelet refractory, meaning they're going to destroy those platelets as soon as they get a transfusion. So you have to give HLA matched platelets. And that's just another picture of where the gene is located. So HLA inheritance is autosomal and it's codominant. So you're going to get a haplotype from each of your parents. And I like this picture. It shows basically each child is going to get 1 haplotype from mom and 1 haplotype from dad. So siblings are 25% likely to have the same type as another sibling. Skip through some of these slides. Another thing you need to know about HLA, and there might be questions about this on the exam, there are some disease associations relating to HLA. So the most common ones are ankylosing spondylitis, which is B27. Also celiac disease, DQ2, but it wouldn't be bad to just memorize this chart. Example question, they might give you something like, patient has these symptoms and they had HLA testing performed and they are positive for B27. What could the disease be? And then you would answer ankylosing spondylitis. So HLA typing, how is it performed? So like I said, it used to be serological typing. So you would take known antibodies and you'd have them in these little tiny plates, which I'll show what those look like. So you'd have, it's called a Terasaki plate. That was the scientist that developed the method. These known antibodies would be all throughout the plate. You would isolate the patient's white blood cells and then plate them all throughout the plate, and then add complement and then see which cells died. And if you had dead cells, then that meant the patient was positive for the antigens of the antibodies that were in that plate. So basically you have a tray map, like, well, 1 has anti-A2, well, 2 has anti-A3, and then whichever wells had the dead cells, you would know that was the type of the patient. But it was very nonspecific. And the more we get into molecular typing, the more we are seeing that these serological types kind of break out. They're not specific enough. Like B15 has many different, more specific antigen titles. And let me go back to typing. So the main methods that we use now are PCR based. So we have SSP, SSO, and then sequence-based typing. And sequence-based typing is Sanger sequencing, but now HLA labs are now getting into next generation sequencing as well. So we'll start with SSO, which is sequence-specific oligonucleotide probe hybridization. So the Luminex, if you're not familiar, it's a really neat little instrument. It is technically a flow cytometer, but you can get these beads from Luminex and you can basically bind whatever you want to look for on them. So in this case, you have a probe and you bind that to the bead. You will isolate the DNA and then do some PCR to amplify the HLA sequence. And then you will basically incubate that amplified DNA with your probe. And it is labeled with a fluorescent, in this case, SAPE, which is strep avidin phycoerythrin. And then you will run it through the flow cytometer and it will be charged by this laser. And depending on what beads glow, you'll be able to put together what the type is. And you don't have to do this manually at all. They have software that basically sees if this bead is positive, then it equals this type. So there's an algorithm built in. Then we have sequence-specific primers. So these are typically ran on gels. So you'll have like a 96 well plate and each well will have a primer in it. And then you'll add your patient's DNA to that plate and then do PCR. And whichever wells are amplified, you'll basically, for each plate, the manufacturer will give you a legend that says, okay, if well 7, 31, and 60 are present, then B27 is present. But it's a combination of which wells are present. So there is some manual analysis with this, and you will see that each lane should have a line there, and that is your internal control. If the internal control is not present, then that well failed for whatever reason. Maybe DNA was not put in that well. It's possible if you're manually pipetting this that you skipped over a well and didn't add DNA. So basically you'd have to repeat that well to have a valid test. And that could be a question on the exam as well. Like what could cause the internal control to fail? So it could be you forgot to add DNA. It could be that well of your thermocycler is not working correctly. So, and then this shows you too. So you have your internal control up here, and then you have your allele specific line, and down here would be your internal control is there, but there's no amplification. Then we have Sanger sequencing. So this is PCR with DD NTPs. So that terminates after each DD NTP is added. So it runs through a capillary electrophoresis and it is struck with that laser. And then you have an output of a sequence. And then Usually these kits will come with software that will help you analyze them. This is what the raw data will look like. And then it will help you analyze. So you'll see this is what your patient looks like at this region of the gene, and then it will give you a predicted HLA type. And then we start getting into next generation sequencing, which we are in the process of evaluating which assay to use for this. So I don't have any screenshots because we're not currently using this. But it is really nice. Sanger sequencing is very manual, and you can only do so many samples at a time. With next generation sequencing, you can do a lot more samples at a time. So it depends on what you're doing this for. If you're doing a lot of patients or if your workload is very high, then next generation might be better for your lab. If you have a low volume of patients, then SSO or Sanger sequencing might be a better choice for your lab. And I won't go into the antibody because it's not really relevant to the molecular side, but just of note, you can do antibody detection with the Luminex as well. Instead of binding a DNA probe to the Luminex beads, you can actually bind an HLA antigen to the beads, and then you incubate it with your patient serum, and then it will tell you what antibodies are present in that patient. So that was a very quick oh, one more thing that may be on the exam that is discussed a little bit are STRs. So in the transplant lab, STRs are used to track engraftment. So this is for bone marrow or stem cell transplant recipients. Basically, STRs are also used in forensics. So you may have seen this on like forensic TV shows. Before you get a transplant, we will do your STR profile, and then we'll also do the donor's STR profile. And so we will know what your profile looks like, and then the donor's profile looks like, and then usually at 30 days post stem cell transplant, we'll take a bone marrow sample to see if that donor's stem cells have engrafted in the patient and taken over and started producing cells. So the way we do that is you can see the patient up here for this loci, they're positive for 17 and 19. The donor is positive for 14 and 18. So you can see post-transplant, the donor is positive for all of those. So what you want to see is that the donor takes over and you don't see any of the patient's type left. So here it looks like it's kind of half and half. So usually if at 30 days it's not looking great like this, then they'll do a 60-day, a 90-day, and keep watching. Sometimes the patient will need, if they have any stem cells left over at the time of transplant, they'll save those, and that's called a DLI, a donor lymphocyte infusion. So if they see like at 30 days they're struggling to engraft, they might give another transfusion of those stem cells to see if that will help push the patient over into letting the donor cells engraft. And this is all calculated based on the area. On this, when you do this, you'll get a printout of each type, what percentage is of each number, and then you'll do a calculation. So on this, looking at all of these, the average was 37% donor. So we want that to be 100% donor after a stem cell transplant. And that is basically HLA typing in a nutshell. Does anyone have any questions about anything specifically?
Thank you so much. I think it was very nice and short, but it cleared a lot of confusions from for me at least. I have a quick question. The last slide that you showed, it showed some of the like the graph. Uh, so I think these kind of questions are very common because they normally can keep some of the pictures. Okay, this is the patients, this is the donor,
Mhm.
and from few options like which one uh, is the like uh, like or
Useful.
something like yeah something like patients and four options of donor like which one will be matched for the patients. So normally what I we try to see was like try to see which has the highest amount of common uh, figure scenario of the graph. uh, if it was little very different, then we think it may be different, like the HLA will be different. But my question is like these pictures, is it uh, uh, which method it used, like SSO, SSP, something like that, or like all the methods, it's gonna show similar kind of graph. Uh,
So for this one specifically, this is STR is done on the same instrument as a Sanger sequencing. So it's done by capillary electrophoresis.
mhm. Okay, okay. Normally nowadays, is it common to just use the sequencing instead of like probe? Because if we use the SSP, like we need to do the gel run, right? That is little bit tedious. Uh,
Well, the one thing about SSP is you can also, like, some companies will make um these kits for
mhm,
SSP, but you don't actually have to run them on a gel. You can run them in a real-time PCR.
mhm,
So instead of having to wait for the gel, which can be time consuming, the real-time PCR is taking the pictures throughout the PCR.
mhm.
So you actually at the end of like 90 minutes, you have a type already.
Got it.
Um, which I didn't get any for today, but I can post and discard kind of some examples.
Mhm,
When you get a new kit
mhm,
for like SSP or real-time PCR,
mhm,
um, they'll have this chart that basically shows like all the options for HLA types on the left side, and then they'll have what positive reactions you should see if you have that type. So like if you're A2, you should have um well 6, well 12, well 19. And then if you're A37, you should have well 30, 37, 42. And the manufacturers of these plates, they try to look because there's common like the more common types, they try to make the kits in a way that they're not going to be ambiguous. Sometimes you do run into that, like they'll have similar positive reactions, so you can't tell. It could be a B57 or B58. There's not a well that will tell you the difference. So then if you run into that, then you would have to do a secondary test to confirm. So um, but I'll send some examples of those. They're very
Yeah.
helpful if you can see the way it's laid out. Um, but yeah, I'll post some of those in Discord.
Okay. Okay. Okay, that sounds good. Because I was thinking like it is kind of discussions. If anybody has any questions, you can unmute and you can ask or you can write in the chat. But in the meantime, I will try to ask another quick question. That is like I was trying to see other kind of like sequencing thing. So I like few days ago I interviewed the Genome Center because but it is in Manhattan. That's why it's a little bit far from. Uh, that's why I could not take that. But over there, what they told me is like um, like for New York State, it's a little bit, they have to confirm any kind of things using the Sanger sequencing. But they showed like they got some of the other state sample that is not required to confirm by Sanger sequencing, like NGS is okay. Or my question is like regarding when we do the HLA typing, is it also depends on different states, all the rules and regulations, or it is like just using any kind of method will be okay? Uh,
So that is in standard. So HLA is mostly, you can be accredited by ASCI
mhm.
or by CAP. And if you're a transplant service, you have to be accredited by ASCI. So they do have different rules for different things. So for heart and lung and kidney, you don't have to have sequencing. You can just do SSO or SSP if you get a clear type. But for stem cell transplants, you do have to have sequencing. And it can be Sanger or next gen. It just has to be um, basically, if you do SSO, it's not super specific. So you'll get a list of the more common types and then some really rare types that it could be, but it's unlikely to be. So in stem cell transplants, you don't want to have that ambiguity. You need to be very clear that you
Yeah.
absolutely know that this is the type of the patient. So you have to basically go deeper to remove all those ambiguities. But
Yes.
for most organ transplants, you could just do sequencing or SSO. Um, you don't have to do sequencing.
Got that. Got that. That sounds clear. uh, Idea. I think I can see some of the people who I know, like I think next week they will have the exams. So regarding this chapter, like if for MB, I can tell like when I tried to study this chapter, like chapter 14, it was little bit I got confused, but I tried to discuss and try to get some ideas. But regarding SMB, like if anybody has any experience, please share, or if you have any questions, please share um, with us. Um, I had like just to clarify, we had another plan with you regarding the specialist blood bank. Just to let everyone know, Miss Anderson already has the specialist in blood bank, but our one of the member has blood bank. She had knowledge about blood bank, but she is shy to. I requested her to host you because I'm not expert in blood bank, right? So I I know some of the stuff in molecular biology. So I requested her to host the podcast, but she said she's very shy, so she gonna tell me like refer me another her colleague, so that I'm still waiting for her. So when she gonna let me know, I will contact with you. Yeah, and anybody else has any other questions?
Hi, Abu. I have one question to Kerry Anderson. Uh, my question is how do you interpret the STR base typing? You know, for STR based fragment analysis for donor and recipient. You know, you said after 30 days or 40 days of donor transplant, basically what you will look to the post transplantation profile.
So it depends on, so basically the doctors will look at how the patient is doing, and they will determine then, sometimes they will take a bone marrow transplant and send that, or sometimes they'll just take a peripheral blood sample, and then we will actually cell sort into B cell, T cell, and granulocytes. And then we'll do a profile on each of those. Because sometimes um, when the patient is engrafting, like the B cells will be fully engrafted, but then the granulocytes will not. And sometimes they'll look at the CBC count, like if they look at the CBC and the neutrophils are very low, they want to see, well, the neutrophils might be low, but are the T and B cells engrafting? Just depending on patients, sometimes the different cell lines engraft at different times. So in that case, this one would be, if you're looking at just one, you're looking at bone marrow. So you're looking at a mixture of all of the cell types. But if you're looking at cell sorted, then you'd have 3 post-transplant, and then it'd be depending on what cell line you're looking at. But you are looking at here, you have a list of the area basically under each peak, and then you're calculating based on that area. So like this one, for example, they're 34% donor based on the peaks. And this one's 38%, this one's 43%. And then you can't calculate percentages if it's not an informative loci. For this one, for example, the patient was 7 before, but then the donor was 7 and 12. So we don't know if this 7 is from the patient's original type or the donor's type. So we can't do a calculation for this one. It's not informative. Did that answer your question?
Yes. Thank you. Thank you.
As it is a discussion part, I also like to add, my question is like STR or VNTR. In both of the cases, it can be used to check the post transplantation HLA, like percentage, or just STR is okay. Like,
so you can't really use the HLA type to check for engraftment, because your donor should be the same type as your patient. So it wouldn't be informative to look at that. So that's why we use STRs, because STRs are so polymorphic. They're usually different between the patient and the donor. Even between siblings, it's usually very different.
Ma'am, do you have any questions? Ma'am, Dr. Amna. Yeah.
Hi, Kelly. Thank you so much for the presentation. My question is not a question actually, because I missed the nomenclature of the HLA using the DNA-based techniques. So if you can just say that again really quickly.
So back up to that. So and this is very confusing. This is called the first group. So this could be, this is the gene. So this would be A, B, C, DR, DQ, DP, whatever gene you're looking at. And then the first group is considered the allele group. So if you're looking at like kidney or organ transplants, you're only required to have the first group, or for platelets. So you just need to know the patient is A1, A2. But once you start doing molecular, you get into the different fields. So each field is going to be separated by a colon. And the second group will be a very slightly different. It is a different protein, but it's very similar to the first allele group. So an A0101, it would be the original gene that was found. And then if you have a slightly different SNP found that causes a protein difference, then it would be an 0102. So they are named in the number that, or the order that they're found. So this would be a SNP that causes a slightly different protein of the first one. And then basically the third field would be a SNP, another SNP that causes some kind of substitution within the coding region. And then the next field would be a SNP that causes a difference in a non-coding region. So it doesn't change the actual antigen at all. And then the last thing, so some may have a number here, most do not, but they will tell you something about the change in expression. So N would mean null. So if they have that type, then they will actually not have an antigen. And there's also L is another common one, which means for some reason they just have a lower expression. S is for soluble or secreted. So it could be on the WBC cells, but it could also be soluble. So it's in solutions. And then Q is basically it's kind of unknown what that change does to the antigen itself. So they found a DNA change, but they're not sure how that affects the actual antigen.
So just to continue my question, for the third field, which is 01, it stands for like a synonymous mutation, right? So it's just to show us that we have a synonymous mutation, but we don't know what it is, right? So like 01 or 02, do they stand for something specific?
So 01 would be the original one. So once you find another one, then you're going to have 02 would be the second one found. 02 would be the original sequence.
01 will be the original and 02 will be any deviation from that.
Right.
So what about if we have an unsynonymous mutation?
So that would be a change in this area. If it changes the protein expression, then it's going to change the second field. So the third and fourth digit. This
Okay.
is a change in the coding region, but it's not altering the protein. This is a DNA change in the non-coding region, which is not changing the protein as well. So both of these are basically silent changes.
And for the fourth field, which is the non-coding, it's always 02 or
It would start, so 02 would be the first change that was reported.
okay, and 01 will be the original.
The reference.
Okay, now I understand.
Yes, it's very confusing. And usually you're not looking at these two. You're looking at the first two fields. It's really only when you get really into this and want to start reporting, like you think you found a new allele or a new change, that you're going to get into all this.
But we can't have like a 03, right? It's either يعني 01, which is the original, or 02, which
No,
is
you
a
can have a
deviation from
03.
that.
So if there's a different one found, then they would just keep going up in number.
Okay, so each number will be like will indicate a specific change.
Mm-hmm. It would be a different change. Yeah. So if I found a different one, a different synonymous, then it would be 02, and then someone else found another one, then it would be 03, and it just keep increasing.
And this I can find in a database or something?
Yeah. So HLA is kind of confusing because it's trying to be international, but everyone uses kind of different standards. So if you go to the World Health Organization, they established a nomenclature, so everyone should be using that nomenclature. The NMDP website has really good, let's see if I can pull that up real fast. They have really great resources on like haplotype frequencies. Let's see, 1 second, let me find that. Change my screen that I'm sharing.
Hello.
Oh.
Hello.
Yes, Dr. Mohit, yes.
Yes. Anderson, I have some few. Actually, we do HLA typing in our lab, but we just started about this disease association, like celiac disease and all. But we are confused about the interpretation. Are you working with the interpretation part of the disease association?
So in our lab, if a doctor wanted that test, they would just order like B27 and we would say it's present or it's not present. And then the doctor would deal with the patient side.
So you will not say whether it's, for example, B27 or a specific allele is intermediate or risk associated or low risk associated, like high risk or low risk. You will not tell.
We wouldn't. I mean, once something is clinically relevant and enough that doctors would order that, then they would order whatever, like B27 or B57,
Yeah.
and then we would just say positive or negative.
Okay. Okay. And there are some. I want to tell to all. Even there's a pharmacogenomics also they use HLA typing. For example, abacavir drugs for HIV. Abacavir drugs for HIV. They do HLA 57, right? HLA B 57. Yeah. Abacavir and carbamazepine. HLA 1501. Yeah. HLA 15B01, I think. So even pharmacogenomics also they do this whether drug is having any risk associated is there or not. They will do the HLA typing, right?
Right. We would have that B57 very commonly ordered by the doctors.
Yeah, that is for HIV, like antiviral therapy they use.
Yeah, and how the patient will react to that medication.
Yes. Yeah, most it might be in SMB they have asked some question like about this what is the HLA for these drugs, which they have asked some questions.
Can you see this chart?
Yes.
We can see that.
So this is something we use in the transplant lab a lot when we're looking. It breaks down by ethnicity, the common types. And so it's inherited as a haplotype. So it can help you. It's basically giving you the rank for each combination. So it can be very helpful when you're looking at HLA runs. But the NMDP website does have a lot of information about common haplotypes and stuff like that, like how to do the Hardy-Weinberg equation. And so lots of nice PDFs available. And that's under bioinformatics on the Be the Match NMDP website.
Okay, that's very interesting.
I have a question. Hi, it's Amna here. Thank you so much for the nice and clear presentation. I am not coming from clinical side of the lab, so I would like to know from the time you take the sample from the patient till you give them the results, how long does it take? And afterwards, so once you're done with the transplantation, do you do it regularly or like what's the criteria for that?
So HLA, once it's done, your DNA shouldn't change unless you get a stem cell transplant. So for most patients, you get one sample, you do the HLA type, and then you're done. And it depends on what type. So at my lab, we're the organ procurement lab. So if you are doing like procurement typing, we had to have that entered onto the UNOS website within 6 hours. So you have to go very quickly. But for most patients, like heart, lung, kidney, we would get the sample, we'd do a full the typing and an antibody profile. So we want to know if the patient has any antibodies so that when we put their type in, we can tell them this is their type, but you also have to avoid these types because they have antibodies to those. So it just depends on what method your lab is using. Like our lab primarily used SSO for all the organ patients. So it might take a week to have everything done because you're also going through, you have like a supervisor review your run, and then you have a medical director review your run to make sure everything looks acceptable. But if you're doing sequencing, it could take much longer. So if you're doing Sanger sequencing, sometimes like the standard primers that come in kits, they don't, you might still have ambiguous typing. So sometimes you have to order special primers to rule out certain alleles, and then it would take much longer. So sometimes like the bone marrow patients, they wouldn't have a full type reported until like a month later if we had to order special primers.
Okay, thank you. Thank you so much.
And those who have exam very soon, I think these discussions help you to clarify your understanding. I will suggest you to just also, I think in the slide there was one concept that I just saw, was like noticed what was HLA null. Try to get some concept on that also, I will suggest. So I think HLA null means the protein is I think in the N means alleles that have been shown not to express. Can you please little bit elaborate? I think it will clarify a lot of people's concept about HLA null.
So there can be multiple things that causes an antigen to be null. It could be there's a mutation in like the promoter region, so that protein will just never be sequenced or never be made. There's lots and lots of reasons why something might be null, but basically for some reason it will not be translated, so a protein will not be made.
Got that. Got that. Okay, so if anybody does not have any other questions, we will end here. Thank you so much, Ms. Anderson, for your nice presentation. Maybe next week we will try to discuss with our another senior member regarding like sequencing, or with Dr. Amram or Dr. Farooq. I am still like processing with my study, my research, and my work and everything. So just stay with us, connect with us, and thank you so much, Ms. Anderson, for presenting, and thank you so much all the participants who joined. I really appreciate your time, and a happy study, and best of luck for your exam.
Thank you. Thank you, Abu.
Thank you. Thank you. Yeah,
Thank
take
you.
care. Bye.
Bye bye. Bye. Have a nice day.